Isolation of DNA is the first step towards the process of recombination DNA technology. In isolation of DNA, lysozyme, cellulase, chitinase, etc. is used to break down the cell membrane of the cell. Then to remove RNA, ribonucleases is used, and to remove protein, proteases is used, and thus the DNA is isolated. After the isolation of DNA, DNA is to be cut at specific sequence. Restriction enzymes is used to cut the DNA at specific sequence. The then purified DNA molecule is incubated with restriction enzyme to cut it at the specific sequences. Then the source and the host DNA both are cut, and then both are kept together and mixed with ligase so that it attaches, and then recombinant DNA is formed. This recombinant DNA is then subjected to amplification through polymerase chain reaction. In this reaction, two set of primers with DNA polymerases is used to amplify, to replicate the more and more number of DNA fragments. Once that is done, this is to be inserted into the competent host, that is insertion of rDNA. The host is made competent by the treatment of divalent cation, and after that various tools like gene guns, micro injections, etc. is used for the insertion of rDNA. After that, obtaining gene product, the product so formed out of rDNA is to be obtained. For this, bioreactors is to be used because the product which is being formed is on a very large scale. So bioreactors are to be used. Bioreactors are of two types: stirred tank and sparged stirred tank. In stirred tank bioreactor, they are cylindrical and curved at end, so that, and stirrer is there so that a proper mixing of oxygen can occur. Stirred tank bioreactor composed of 4 units, as you can see in the diagram: agitator unit, oxygen delivery unit, temperature control unit, and foam control unit. Once we have obtained the gene product, then we have to do the downstreaming processes. The downstreaming processes are nothing but separation and purification processes. These are done in order to remove any impurity or any unwanted material from the gene product. Once this is done, quality check is done, preservatives are added, and clinical trials are done in case if the product is of medicinal importance.